Hello, everyone. I'm Tim Karachi, and I'm the Vice Chair for the Department of Medicine, acting on behalf of Dr. Crowther with presenting chairs rounds today on January the 9th. Hope everyone had a happy holiday and is ready in earnest to get back to work. I'd like today to introduce everyone, if you don't already know, Dr. Elena Verdu. She is a professor in our Department of Medicine. I'm very proud of all of her accomplishments. And she is the director of the Farncombe Family Digestive Health Research Institute. She's been a part of that, I think, since in the leadership since its inception and been vital to the success of the Institute. She holds a Tier 1 Canada Research Chair in Microbial Therapeutics and Nutrition and Gastroenterology. And she has a research that focuses on the complexities of gut microbiota and its impact on gastrointestinal health. And as you can see from the title of her presentation today, she does have a very specific particular interest in conditions such as celiac disease and inflammatory bowel disease. And we're going to hear about celiac disease from discovery to clinical practice. So with that, I will hand things over to Dr. Verdu and looking forward to this presentation.
Thank you very much, Tim, and thank you for inviting me to speak about one of my favorite conditions in gastroenterology, that is celiac disease. And I have to disclose, I use the title, I self-plagiarized from a recent issue dedicated to celiac disease in gastroenterology that I'm very proud of, and I co-edited with Dr. Green in Columbia University. So what I would like, and Happy New Year to everyone. What I would like to do today is to review these objectives. First, speak about immunopathogenesis and also epidemiology and the clinical presentation of celiac disease. But what I would like to do throughout the talk is to emphasize how these fundamental discoveries actually have translated into new diagnostics and into making a difference into the management of these patients. And then, of course, at the end of the talk, I would like to introduce the work we do in the lab and in the Farncombe, which focuses in microbial triggers. So to start with a little bit of history, I think we have to mention this fantastic clinical observer and translation investigator that was William Dicke. He was a pediatrician. This is the first page of his thesis, but he's the one that actually observed that in children, being a pediatrician, symptoms of vomiting, steatorrhea, and anorexia, they disappear following and followed by recovery of weight and growth after a wheat-free diet. He didn't stop there. He went on to do a challenge and to see that these symptoms were reproduced or recurred if wheat, rye, and bread was reintroduced. And he went on to do studies in the bench where, through a series of studies, he actually identified that it was the proteinaceous fraction in the cereals that was the injurious component. And so this is what I will refer to when I speak about gluten. It's the over-encompassing term that, in fact, is not very accurate, but for simplicity, I will use it because it encompasses a myriad of proteins that are immunogenic for celiacs and that actually belong to the prolamine fraction of these proteins. And so this was an incredible discovery. Already the Greeks knew about these symptoms, but this is what allowed for the first and only treatment that we have in celiac disease so far, which is a gluten-free diet, very strict and for life. But it led immediately in 1 or 2 years to the discovery of the lesion in the small intestine, the atrophic lesion, the blunting of the villi by Polly. And this was in patients undergoing laparotomy, and later on by the development of the precursors of endoscopy, the peroral instruments of duodenal biopsy that confirmed the presence of biopsy. And it was not until 1992 that we obtained a score system, the Marsh system that we use today to grade the lesion. So let's fast forward 75 years later. What have we learned about this disease? And in fact, for several decades, there was a stagnation in celiac disease where we considered this a food intolerance that was cured after removal of gluten-containing foods. Well, we now know that this is an immune-mediated disease. It is driven by CD4 T cells that are specific to gluten. We know that it's an autoimmune condition. This is not an allergy. This is not a food intolerance. And we know this because we have discovered the autoantibodies, anti-tissue transglutaminase 2. I will talk more about this because there is specific association with MHC class 2, and this is mainly DQ2 for more than 95% of the patients, the rest DQ8. We know also that resolution is slow. It's incomplete. We are happy if patients on a gluten-free diet achieve a villus to CRIP ratio of more than 2.5, but they rarely return to what a normal individual would have. And we have new immunological evidence that the memory cell response, both B and T, display, although these are non-proliferative cells, they express certain activation markers that are evidence of chronicity. And the other myth that has been debunked is that this is not a disease of European people of European descent. It affects most ethnicities over the lifespan, and this is a systemic condition that the internist should know about. So what are the symptoms of disease? Of course, in children, pediatricians are very good at this. They know that individuals with diarrhea and failure to thrive, usually they are tested for celiac disease. But in adults, and we are seeing more diagnosis in the adult population, and we can talk about that during the questions, the presentation is usually non-classical, meaning that we can have gastrointestinal presentation with typical abdominal pain, diarrhea, bloating, but we can also have patients that are constipated or patients that do not have any of these symptoms and monosymptomatically have an unexplained transaminemia, so elevated liver enzymes. They can also present with anemia or with skin abnormalities or infertility. All of these are, I'm not going to read them one by one, but neuropsychiatric disorders as well. And so all these subspecialists need to think on a potential diagnosis of celiac disease, especially if associated with other conditions. And these are all immune-mediated or autoimmune diseases. And just to name a few, thyroiditis, type 1 diabetes, these are all at-risk groups where we do need to think about celiac disease because they may not present with gastrointestinal typical symptoms. And so there has been a change in the global prevalence. And this has also shown that there are some inequities in celiac disease diagnosis. So on the left, you have a graph from predominantly Europe and US studies showing this increase in prevalence. And we know this is true increase in prevalence because some of these studies have been done in, for example, biobank samples over a period of 40 years using the very specific antibodies that we use today. So this is not just better diagnosis because we have a better diagnostic procedure. But what these studies are also showing is that we are starting to see celiac disease worldwide. And the latest studies show global prevalence of 0.7 to 2.9, depending on the geographic areas. We see more cases in Africa, Asia, Middle East, places where we thought we would not find celiac disease. And we also know now that native population of the Americas, and this is a paper in South American and Mexican populations, but we also know from our clinical practice that North American indigenous people can have celiac disease because they have the genetic background. Now, in the US, and this is going on into inequities, it is known that being diagnosed with celiac disease is negatively correlated with being Black, Latino, or with a median social deprivation index. And this is paralleled in Canada. This is a very nice study from University of Calgary, showing that while testing rates in the province of Alberta have remained stable, the incidence has increased, and it's higher in urban areas and in females. And this was unveiled, actually, the Canada Celiac survey in 2023 that shows that most of the diagnosed celiacs in Canada are white, and they are from higher income or urban. And so we know that celiac disease occurs in other ethnicities, so we are failing really the diagnosis of these patients. So there is an increasing burden of celiac disease. And of course, this has to do with not diagnosing this disease properly. And depending on geographic area, there is about 50% or even more in some areas of undiagnosed cases. So most celiacs, or at least half of them, are undiagnosed worldwide. And the prevalence is increasing. And these individuals continue to seek medical care. They have long-term complications, many times, especially in females, but also in males, related to bone health. There is decreased quality of life. Of course, this increases healthcare costs. When we make a diagnosis, there is still a burden of celiac disease that has to do with different factors, with bad management or poor follow-up that leads to nutritional deficiencies, potential frequent contaminations. We are going to speak about this, mention it quite often. But also there is a psychological and psychosocial burden and a cost burden that actually is put on the patient because the patient is the one who has to do the gluten-free diet, and there are many limitations to this treatment, although is the best we can do for the patients. So the factors, I think, potentially responsible for this increasing burden has to do with the increase in prevalence, but also that most of these patients are not going to be diagnosed, and we have to get better at that. But also environmental factors that are associated with a general increase in autoimmune disorders, but for celiac disease could also relate to the fact that certain populations are changing their diets, and they are eating things like burgers or pizza when traditionally they didn't have access to this and their diets were low in gluten. So moving on to the immunobiology of the celiac lesion, I love this cartoon because when I share it with my gastroenterology colleagues that work on IBD or in IBS, and their cartoons are full of arrows that have question marks everywhere. And in this cartoon, you will notice there is just one little question mark here that relates to what is activating these innate cells. But all the adaptive immune response in celiac disease is very well characterized. And you will allow me, I know this is very complex, but to be more didactic, and I will put in sequence the key immunopathology of celiac disease. Of course, these things weren't discovered in this order. They were put together as we put together a puzzle, the little pieces of a puzzle. So the first one I want to refer to is the fact that gluten proteins are resistant to digestive proteolysis. So our enzymes, pepsin, chymotrypsin, cannot fully digest gluten, and that has to do with its chemistry. Now, microbial proteases are a different story, and they can cleave some areas that our enzymes cannot, and I will come back to that. But it is the resistance to proteolysis that leaves these peptides that are going to be taken up into the lamina propria, and there they encounter an enzyme that is tissue transglutaminase 2 that in celiacs is activated, and it's extracellular. The source of this enzyme can be the endomysium, but also epithelial cells. And so this discovery was key because tissue transglutaminase 2 plays two roles. One, it deamidates gluten, and by deamidating gluten, it makes it more avid to the MHC class 2 association, and therefore it promotes a stronger CD4 pro-inflammatory T cell response. But the other role of TG2 is that it becomes the autoantigen. And this has allowed for a very specific diagnosis for celiac disease. So the way we know that it's the autoantigen is because TG2 and gluten form complexes, they are taken up by the B cell receptor, and this B cell will present the antigens, both TG2 and gluten, to the CD4 T cell. And that's how we get anti-tissue transglutaminase 2 antibodies that we use for the diagnosis. Now, the last step is the cytolytic effect of IELs, intraepithelial lymphocytes, that kill epithelial cells, and that's, together with the CD4 T cells, what causes atrophy. And because these cells are part of the innate immune response, they are not gluten-specific. And here is where there is quite a bit of immunological investigation onto how are these cells activated. Because if we don't have this response, we can have the serological response and what we call celiac autoimmunity, but we won't have enteropathy. So how did we arrive at this very deep understanding? And I really want to emphasize the role of very few investigators that working together could put together this puzzle. So as I said, these steps were not discovered in this way. Probably the first important discovery was observation was by Ludwig Solid in Scandinavia, where he identified that celiac disease associated very specifically with HLA-DQ, alpha-beta heterodimer. And he went on to discover or to show that this was mainly HLA-DQ2, but also later on DQ8. And at that time, and this is the early 90s, we also knew that patients with celiac disease had antibodies, IgA antibodies in the serum against gliadin and the endomysium. So this was a clue that perhaps this was not a food intolerance and that there could be some kind of autoimmune pathogenesis. And so it was Detlef Schupan that actually, through a series of studies, discovered that when you block tissue transglutaminase 2, which was part of the endomysial candidates, then these antibodies disappear. So that's how tissue transglutaminase 2 was discovered as the main autoantigen. And in that paper also, it was shown that gliadin, which is a subfraction protein from wheat gluten, is the preferred substrate for TG2. But that's all we knew. We didn't have any clue on what happened with this interaction. And that's when, at the same time, I'm quite sure Ludwig Solid and Fritz Koning were paying attention to this, following this research. And what they found was that TG2, what it does is to deaminate gluten, and this is associated with enhanced T cell reactivity. And I love this picture. It's the original paper of Ludwig Solid, and we are still doing this kind of experiments, where if you isolate PBMC cells and then you incubate them with tissue transglutaminase 2 and you deamidate gluten, then PBMCs or gluten-specific T cells are going to proliferate much more. And so remember this because we have used this in future experiments. So at the same time, Chetan Khosla, who is a biochemist, was doing studies on why these peptides are present in the gastrointestinal tract when we eat gluten, why they are not digested. And so what he did was LCMS studies where he incubated alpha gliadin with one of the main proteins in gluten with many digestive proteases. And what he confirmed was that all these peaks are peptides that are highlighted here in the alpha gliadin sequence. And I want to call your attention to the one in red, which is the 33-mer, because you see that there are many Q and P sequences. These are prolamines and glutamines that are not able to be cleaved by our enzymes. Now, bacterial enzymes can chop this part. But this is why we have these peptides that cannot be further digested. And Ludwig immediately collaborated with Kosla because he said, wait a minute, these P and Q areas are the site of the amidation of TG2, which we know make T-cell responses more effective. So this triggered a structural basis, the study of the structural basis for the deamidated gluten and the binding to HLA-DQ, which he had previously discovered. And basically what is happening is that tissue transglutaminase 2 is deamidating these glutamine residues, introducing a negative charge. It becomes a glutamic acid. And this negative charge makes the binding to certain pockets in the DQ2 or later on in the DQ8 MHC class 2 molecule very efficient. So then this interaction between antigen presenting cell and CD4 T cell is very strong, and that's when we get clonal expansion and proliferation and production of interferon gamma and IL-21. And so we can go back to this cartoon slide. And the importance of these were all basic studies with no industry behind to push them because we didn't have a drug for celiac disease. But suddenly now we have this richness of mechanisms, very specific steps that are all druggable. And so where are we with drug development in celiac disease? And this is a very active area. I'm not a clinical trialist, so I'm not going to speak individually of these studies, but just to put it in context with what I said. So, for example, the lack of proteolysis of gluten is being tackled by gluten digestive enzymes that have been engineered by AI or that are a mixture of microbial enzymes to target those areas that we cannot digest. The adaptive immune response that I described is being targeted by vaccination approaches. This is more a retolerance, a tolerance induction using those peptides that are immunogenic. We also have TG2 inhibitors. We mentioned this is a key enzyme, this deamidation that increases the immunogenicity of gluten. And this has been recently published in New England Journal of Medicine, and the drug is moving forward to a phase 3. And then finally, we have antibodies against IL-15 to tackle that innate immune response, so the cytotoxic response and the activation of epithelial cell killing, which is mainly driven by this cytokine IL-15. So now to the last part of the talk, the third part, where I would like to give the rationale of why we are interested in triggers, in microbial triggers in celiac disease. We know that gluten is the main environmental driver, but other cofactors have to act as triggers or as potentiators of the immune response. And the reason we say this is because we do have the necessary factors to develop celiac disease, which is, of course, exposure to a gluten-containing diet, but also the presence of genes, HLA, DQ2, and DQ8, as I mentioned. But these are very prevalent genes. 40% of worldwide population have these genes, and only 2 to 3% are going to develop the disease. Now, this, tied with the increasing prevalence that I mentioned, is crying out for additional cofactors. And in the area, we've always suspected several categories of cofactors, but because we are working in the pharmacom and we are interested in microbial host interactions, of course, we are interested in microbial triggers. But this also has some clinical rationale. There are post-infectious case reports of celiac disease, especially adult onset, that are described. Patients with celiac disease, active or non-responders, they have antibodies against microbial components, and some of them are anti-Pseudomonas, florensis, or aeruginosa, and I will come back to that. The prospective studies in children that have been completed and followed up for 10 or 14 years, including the TEDdy study, Predict City, they all show that infections in early age are a risk factor, especially when those infections occur at the time of a high load of introduction of gluten to the diet. And then, of course, in the last 20 years, the association with microbiome changes. And so I'm going to focus a little bit more on this. So this is a difficult table to navigate and read, but what I want you to focus is on the color coding. So here you have all the studies that we reviewed and put together on all cross-section and longitudinal studies that show some changes in the microbiome in celiac disease. And here on the left, what you have is the different taxa. In red, it means that those studies are reporting an increase in that specific taxa. In that case, for example, where I'm pointing is proteobacteria, or in blue, it means that those studies have decreased abundance of those taxa. Now, the first thing you see when you look at this table is that there is a lot of white, which means although we have associations that are being reported in individual studies, they are not always congruent. Although some taxa are more congruent than others, like Enterobacteriaceae, Proteobacteria, Neisseria. But basically, these are clinical associations. The other point is that most of these studies are areas, and they give us little mechanistic insight. And so we are lacking biogeographic information. Remember, this is a condition that affects the duodenum or jejunum. And if we want to do mechanistic studies, what we need is to do culture and isolates. So this is why we performed this systematic study a few years ago, where we obtained samples from D1, from D2 and D3. We obtained also duodenal aspirates during endoscopy and stool samples. And these were from individuals newly diagnosed with celiac disease, so with active celiac disease consuming gluten, and from controls. And the main result that I want to show you is that this is showing the beta diversity, the different ways that the microbiome is clustering. And you can see that all the green samples are the biopsies, the blue are the aspirates. So biopsies D1, D2, D3 mixed there, and the feces are in brown. And its feces are really profiling or clustering quite differently than the duodenal samples. And this is confirmed by the statistics, although having or not having celiac disease can give you a different microbiome profile. Location is the most important factor to determine whether the microbiome clusters in one place or the other. So if we move on and we just concentrate on D1, D2, and D3, we also see the same thing. Yes, here maybe the fact that we are looking into the duodenum, having or not having celiac disease seems to play a higher role, but location is still very important. And let me just illustrate that with Neisseria. Neisseria is a taxon that was shown in that list of papers to be more congruent than other taxa. But here we only see it significantly increased, and light blue means higher abundance. In D2, we don't see it in D1 or D3. And rarely we get this information in clinical studies of where the sample has been taken. So we were able to do a predicted analysis of the microbial gene pool that we had in D1 and D2, D3. And of course, we focused on proteases because this is what is impaired in the mammalian capacity to degrade gluten. And what we found was that many proteases that are predicted to degrade those unions of proline and glutamine are decreased in D2 at least in individuals with celiac disease. Of course, this is a prediction based on the gene pool. So for this reason, we validated this with mechanistic studies in the lab. And what we have here are germ-free mice that can be colonized with these microbial communities. And so we colonized these mice with healthy control bacterial communities from the duodenum. And we divided these donors into those that were heterozygous for DQ2, so with some genetic predisposition, or those with negative genetic predisposition. And then, of course, all celiac were heterozygous for DQ2. And we did gluten degradation experiments in the lab. And indeed, what we see is an impaired capacity of those microbes in the mouse that was colonized to degrade gluten challenges. And we see kind of, it's not statistically significant, but it's interesting to see that the microbiome of the controls that did not develop celiac disease but have some genetic predisposition have very good capacity to degrade gluten. We did this ex vivo as well. So again, blue means lower here. And these are tripeptides. You see the prolines and the glutamines that we test for cleavage, and there is lower cleavage of these areas with intestinal contents of the mice that are colonized with a celiac microbiome compared to the other two groups. So this is telling us that the microbial community of the small intestine has a lower capacity to degrade gluten, but we don't know what the exact outcome of the metabolism is, and we don't know whether this has any capacity to influence the CD4 T cell responses. And so this is when Alberto Caminero, who is now assistant professor at the Farncombe Institute and in the Department of Medicine, came as a postdoctoral fellow to work on gluten metabolism in the GI tract. And so this is what I mentioned before. When we consume gluten, we have a certain proteolytic resistance that leads to peptides like the 33-mer. This is not the only proteolytic resistant peptide. It's just a well-characterized peptide that contains the immunogenic sequences that most DQ2 individuals react to. And so what Alberto started to show was that the upper GI tract, both of healthy controls and of celiac patients, do contain microbes that are very avidly degrading gluten. And I love this gluten agar plate. This is the protein source is gluten, and you see the hydrolytic halo of that taxon that is actually eating avidly gluten. Now, of course, this doesn't tell us exactly what that degradation is it partial or not? And so this is why Alberto embarked in collaboration with the Biochem Institute into similar experiments as Chaitan Khosla had done, but in this case, not with mammalian enzymes, but with digestions by isolates that we got from celiac patients. And so here the 33-mer was incubated with Pseudomonas aeruginosa. You have here the 33-mer, the sequences that are immunogenic, and you see that after 15-20 minutes of incubation, there is no degradation to amino acids. Yes, there is some chopping. The Pseudomonas is eating gluten somehow, but there is no full detoxification. Now, he did more experiments where he added other bacteria, and then he got different types of peptides. And he also, in collaboration with Michael Surette, tested this same metabolism, but with a mutant of Pseudomonas aeruginosa that does not have elastase. And we did not get this production of peptides. So we know that the main enzyme in Pseudomonas aeruginosa for the production of these peptides is elastase. So to answer the question whether this is immunogenic, we did more or less the same experiment of Ludwig Solid, where you deamidate gluten, or in this case, a 33-mer. And when you deamidate gluten and you take cells from celiac patients that have been challenged with gluten, you will see in an elite spot response, for example, this increased immunogenicity. This is the positive control of the assay. So we then repeated that, but using the peptides that had been produced by the Pseudomonas digestion, because we wanted to know whether those shorter peptides were immunogenic. And they were highly immunogenic. So Pseudomonas does not detoxify. However, if we now continue the degradation with other bacteria, like lactobacillus, many of those peptides did not induce the pro-inflammatory immune response. So what did this study conclude? That the duodenal community that a patient or an individual harbors impacts the net outcome of gluten degradation and of immunogenicity. And in a celiac patient, this could make a difference. Now, moving on to the metabolic capacity of the microbiota. I mean, it's enormous. It can be a more efficient metabolic organ collectively than the liver. And so other important metabolism, it's not only gluten metabolism. At the same time we were doing these studies, Harry Sokol in France and Bruno Lamas, his PhD student, were working on tryptophan metabolism by the microbiome. Now, tryptophan, as you all know, is a essential amino acid. It has this indole ring here that can be metabolized by microbial enzymes. The metabolism is combined with host pathways, so the kynurenine pathway and the serotonin pathway as well. But we were very interested in this microbial degradation because this would produce indoles that signal through the aryl hydrocarbon receptor, which was implicated in the maintenance of gut health and homeostasis. And what Bruno had shown in Harry Sokol lab was that alteration of this microbial degradation due to the presence of also a dysbiosis in IBD is implicated in pro-inflammatory pathways through a lower activation of the AHR receptor. So when he finished his PhD, he came as a postdoc to my lab to work on our clinical cohorts. And so what he did was to study tryptophan metabolism in active celiacs, controls, and in patients with celiac disease 2 years after the gluten-free diet. And we did metabolomics, AHR activation, and also he took advantage of our genetically predisposed mouse models like the DQ8 transgenic model. And so these are the results of the metabolomics targeted to tryptophan metabolism in celiac patients. And you can see that there is a decrease of AHR agonists in the GI tract. There is, interestingly, decreased tryptophan, total tryptophan, but the renin pathway seems to be increased. And this makes sense. If one is depressed, then the metabolism goes more to the other arm. And in terms of functional activation of the AHR receptor, and this is done in a reporter cell line, the contents of the intestine of celiac patients have less capacity to activate the AHR. And this is somehow partially restored after the gluten-free diet. And this is confirmed by gene expression in the duodenum. These are all genes that are important in the AHR pathway downstream. Now, IL-15, as I mentioned, this is not part of the AHR pathway, but it is a typical cytokine involved in the intestinal epithelial cell, intestinal lymphocyte activation, and this is increased in celiacs, and as you can see, it decreases, but not so much after the gluten-free diet. So there is residual inflammation in these patients. He then moved to investigate whether he could rescue this in animal models, and so he used our transgenic DQ8 mice. These mice have to be sensitized with a bacterial antigen like cholera toxin, and with gliadin, and then challenged orally to induce enteropathy. And then he subjected these animals to a lower tryptophan diet or a supplemented tryptophan diet. And he also treated some of these animals with 2 lactobacillus reuteri that were known in vitro to produce indoles, so to metabolize tryptophan for the production of indoles. And what we found was that, of course, AHR activity is mainly activated when we give both, when we give the substrate and we give the bacterial metabolizer. But what was interesting is that villus to crypt ratio, so the enteropathy, was rescued both with tryptophan and with a combination. And we thought this was due, based on some microbiome results we got, to the fact that giving the substrate already stimulates the endogenous microbiome to induce this tryptophan metabolic capacity. So this gave us an idea that maybe in the area, everybody is trying to find what is the exact indole that is anti-inflammatory. And this is mainly in the area of IBD. But with these results, we say, wait a minute, maybe all we have to do is to deliver free tryptophan to the small intestine. And so we conducted this proof of principle study in healthy controls, where the objective was to see, can we activate the indole pathway and the AHR activation just by giving free tryptophan delivered to the small intestine? And so we did this randomized double-blind placebo-controlled trial. We gave 3 grams of tryptophan or placebo. These were enteric-coated capsules. And we studied the metabolome of tryptophan in aspirates, in urine, in blood, in stool. And also we did AHR activation studies, as well as we monitored symptoms, both GI and mood related, because of the tryptophan pathway. And so these are the results. They have recently been published. We actually can activate the pathway just by giving oral tryptophan in this manner. There is an increase in total indoles and in some of the subsets of indoles in the L-tryptophan in treated arm. We can see the same in the urine, but we do not see any increase in feces. So we really think that by this type of administration, all the metabolism, or the important metabolism, is occurring in the small intestine, and not much is coming to the lower GI tract. And this may have implications of how people want to intervene using this pathway in IBD when they want to treat lower GI disease. So in terms of activation of the AHR pathway, and these are normal individuals, so they shouldn't have altered AHR activation, but we did see a modest increase in activation capacity with duodenal aspirates, not in feces, again supporting that this is happening in the small intestine. So this is what actually took us to say, okay, then now we have to go and intervene in non-responsive celiac disease patients. Now, non-responsive celiac disease is quite common. This is not refractory. We can discuss it later. Non-responsive means that these are patients that have persistent symptoms despite being on the gluten-free diet, or that respond and then suddenly they become symptomatic again, or they have borderline TG2. So the number one cause for this is that these patients are not completely gluten-free, because it's very difficult to do a zero gluten-free diet. And so we targeted this population with a study that basically mirrored the one that had been conducted by Gaston Rueda in healthy controls. And here we had a primary objective, which was the improvement of celiac-specific symptoms. And the secondary objectives were improvement of GRS and quality of life, but also some objective markers like indole production and microbiota composition. And so this study is still ongoing, but we were able to reach midpoint and unblind with the patients we have. We have 23 that have completed the study. And so what we see is that, of course, we are not powered with half of the patients. Our aim is to go to 50. We see a higher percent of patients that in the tryptophan arm have improvements, meaningful improvements in the celiac symptom index. We see the same trend for the GSRS. The GSRS have similar symptoms of the celiac symptom index, but does not include the does not include quality of life. And so quality of life was also one that was improved in the tryptophan group. So we'll have to wait for the objective studies and the full study to be completed to know whether this is something that we could implement in our non-responsive patients. And so our studies over the past years, they have identified, and it's not only us, there are many other groups working, especially in viral pathogenesis, multiple mechanisms by which microbes can act as triggers of celiac disease. And I mentioned mainly all our work in gluten metabolism with pathogens like Pseudomonas with partial breakdown, but that can, even if present, they can be fully detoxified if there is a community of commensals that can continue the digestion. And so this could be used therapeutically, and especially with the interest now in microbial therapeutics, to use targeted therapies for this mechanism. The other one I mentioned are the potential for indole producers, or as we showed, maybe all we need to do is provide the substrate and free tryptophan in the duodenum. I didn't mention immune tolerance to gluten and modulation by microbes. There's a lot of studies here. Wonderful work by Bana Jabri in University of Chicago. Viral infections in the longitudinal studies have been shown to be associated with at-risk individuals to later develop celiac disease. And so the potential for example, vaccination in early life that could prevent loss of oral tolerance, or a study that we did in collaboration with Dr. Hintel-Leitner in the US, showing that certain components of the duodenal microbiome, not bacterial, like Parabasalia, these are protists, could actually prevent the loss of tolerance that viruses induce. And finally, I didn't speak about this, but Pseudomonas, although we implicated it in a mechanism through gluten metabolism, other investigators have produced very elegant studies showing that certain sequences of Pseudomonas can be recognized by the gluten-specific T cells. And so this is molecular mimicry, and maybe this could be also targeted by some vaccination or immune approaches. And so just to end up some present and future clinical considerations, for me, it's undoubtful that the discovery of TG2 is what allowed for a complete change in the way we diagnose celiac disease. But this also led to accurate epidemiology that brought up all the concepts that we've been mentioning throughout the talk. We know that most celiacs are undiagnosed, that prevalence is increasing, and despite the stabilization of testing, the prevalence continues to increase. We also know that gut microbes can modulate immune responses to gluten and through very defined mechanisms, and this could lead to some targeted microbial therapeutics. We know also that the duodenal lesions are patchy and they can just involve the duodenal bulb. So we have these gray zone conditions of ultra short celiac disease, or even just in the duodenum, that require different, we need to know about them when we get these complex or non-classical cases. We have discordant cases like seronegative celiac disease, or serology but without tissue damage, and that's a potential celiac disease. Others call it celiac autoimmunity. And the fact that treatment has remained dietary, hopefully not for long, but we know that the gluten-free diet has to be very strict and lifelong, and it's not possible to completely prevent contaminations over a lifetime. This creates many burdens, social and economic burdens for the patient, and we need to keep in mind that not all patients are going to fully respond to the gluten-free diet for a variety of reasons. So this is why non-dietary therapies are being developed. And I want to make the point that this pipeline wouldn't have been possible without detailed mechanistic knowledge provided by basic and translational researchers. So basic research is important. And it's not only therapies. We do have new diagnostic methods that are being developed and that could be used, for example, to avoid the long-term gluten challenge that sometimes in these discrepant cases we have to do, and patients suffer by putting them on gluten-free diet, or sometimes they simply don't want to do it during several months of gluten challenge because they get quite sick. And these new diagnostic methods could circumvent this problem. And so with this, thank you very much for listening. And I want to thank all my collaborators and especially the people in my lab. Nothing is done in isolation. And so ready for questions.
Dr. Verdu, thank you so much for that. I'd like to point everyone toward Dr. Verdu's lab. You can find a website, verdulab.com, and it'll describe the extensive network of individuals that she's working with, supervising, and collaborating with, and all the immense work that's going into this. I think your presentation today has really highlighted truly the complexity of the gut microbiota and its impact on gastrointestinal health. And really, that use of what I didn't really know until I read about your website, this notobiotic model, as a way to study this for finding the mechanisms to link bacterial communities and these illnesses with immunogenicity affecting them. And so I think celiac disease, IBD, I think you're probably just scratching the surface. I wonder, the effect of medications that we give to people. I know in the ICU, where we stress ulcer prophylax people with proton pump inhibitors, and how that simply can affect the microbiota on its own, let alone anything else that we do. And just even in that short window of time that we provide those drugs to people, I wonder about, long-term, effects and other things. Any comment on that?
Yeah, actually, Tim, I did my my clinical research thesis in Lausanne on PPIs and microbiome, because it was the hype of the PPI discovery. Omeprazole had just been discovered. And so with David Armstrong, who was there at the time, we conducted several studies where we demonstrated that 4 weeks of omeprazole, and then we did this with lansoprazole, does induce bacterial gastric overgrowth. This is short term, and there are pockets of acidity. So, it's when you have to give the proton pump inhibitors, you should give them. But it also induces duodenal bacterial overgrowth with bacteria that actually are coming from the oral cavity. So in the ICU, this has to be taken into account if you're concerned about aspiration pneumonia, right? Because PPIs are going to induce this bacterial overgrowth. So I mentioned PPIs because that's what you asked for, but with the hype of the microbiome, I would say that today many interactions are being studied, and probably the ones with the checkpoint inhibitors for cancer treatment are the most popular ones. And so we are seeing many interactions, not only at the metabolic, but maybe of efficacy of changes in efficacy of these drugs.
Yeah, I think that's going to be an important field that we'll have to layer onto all the other, benefits and disadvantages of a lot of these therapies for what we're studying them for. I'm monitoring the chat, so if anyone has questions, we certainly welcome them. We've had a really great turnout, approximately 50 people showing up. That's a very large number for our rounds, given how busy everyone is. So really appreciate people taking the time to join today. We have a few more minutes if people have questions. And you have a lot of grad students, I think, in your lab. You have a very busy lab. And it looks like, I'm sorry, I'll stop. Jill Rudkowski has a question. Can you talk about clinical approach to patients with a family history, vitamin D, B12 deficiency, negative TTG?
Okay, so as I mentioned, you have something that is called serologically negative celiac disease. So of course these are patients with negative serology, but they have flat villi. So you would need to investigate if you are thinking of celiac disease. Just ask for an endoscopy, perhaps, to rule out that there is no lesion in the duodenum. Of course, when you have these cases of seronegative celiac disease, it is a diagnosis of exclusion because you need to exclude all other causes of enteropathy that can cause the nutritional deficiencies that you are describing. So celiac tropical sprue, giardiasis, H. pylori, even HIV, Crohn's disease of the GI tract. So it is a diagnosis of exclusion that you have to do. But one easy way, if you want to rule out celiac disease easily without doing the endoscopy, is just genetic testing. We can do this in the hospital, and if this is negative, it's unlikely that it is celiac disease. And so that's one way of approaching it.
Thank you, Jill, for asking that question. Oh, she's got an extra one.
If positive HLA, if positive HLA and you rule out other drug, infection, immune-mediated causes of atrophy, then yeah, it's celiac disease, and then you should put them on a gluten-free diet, which will actually tell you 1 year later, or if the endoscopic lesion has improved, or even the symptomatic, so your nutritional deficiencies have been corrected. So it's a therapeutic proof.
I agree. Super helpful for sure. For sure. I really appreciated your comment and where showing that cartoon, and admitting the question marks that exist. And really that's where the fruit of finding, new topics for study really come from. And looks like you're really uncovering and clarifying those question marks on those mechanistic cartoons that you described. That really helps for clinicians like myself really get a better feel for what's going on, and having you explain it, I think, has really helped people like myself, and I'm sure others on this rounds. We've got another minute or so for other questions if anyone wishes. If not, last words, Dr. Verdu, before we sign off and I get to thank you.
Well, just think about celiac disease, because regardless of the specialty or subspecialty you have, it is a clinical chameleon. And if we don't think about it and we don't ask for the test, we are not going to diagnose it.
Yeah, absolutely. Thank you. Really appreciate that sentiment. David's got a question. Here's one last question I think we can take. If celiac disease is prompted by an infection, should we avoid grain products if we get gastroenteritis?
Oh, well, it's difficult to say, to put someone on a gluten-free diet without a proper diagnosis. In order to put them on a gluten-free diet, because it's not trivial, you would have to make diagnosis of celiac disease. Otherwise, it's not recommended that you give gluten-free diet recommendations to someone without a diagnosis.
Yep. Now you're getting questions from other experts here.
Yeah, because there are issues. There are issues with a gluten-free diet on itself. It's low in fiber. It's not a wonderful diet, the gluten-free diet.
All right. Well, thank you so much. Really appreciate this. Again, if you have questions, I'm sure you can reach out to Dr. Verdu directly. You can find a lot more about this on her website for her lab. The lab is a very busy, active lab. But I'm sure she's looking for other questions if you have them. Thank you again, everyone. This has been a great turnout and hope everyone has a great day. Thank you again, Elena.
Thank you. Bye.